Hi students, how are you? Today we would like to talk about enteroviruses, which are very important pathogens can cause a children's disease. And today's lecture will include 7 parts: virus characteristics, life cycle, epidemiology, transmission, pathogenesis, laboratory diagnosis, and treatment prevention. Now let's move to the first part regarding virus characteristics. This virus belongs to the family Piconaviridae. Pico in Latin means very small, so you can see the very tiny virus particle under electron microscope. RNA is because its genomic material is RNA, so we call it picoRNA virus. The family of piconaviruses contains 5 genera: enteroviruses, rhinoviruses, arbathoviruses, cardioviruses, and hepatoviruses. And here we focus on enteroviruses. And as you can see in the side, the enteroviruses include many groups, including Coxsackie A, Coxsackie B, echoviruses, and numbered enteroviruses. The most famous one are EB71 and EBD68. They caused many big outbreaks recently. So we will continue to introduce enterovirus later on. Let's continue the characteristic of enteroviruses. Like other piconaviruses, this virus is naked and composed by capsid proteins. And inside of the capsid, there is an RNA we call positive-stranded RNA. Why it is positive-stranded RNA? It is because the RNA itself can serve as messenger RNA, directly translate into viral proteins. This virus replicates in cytoplasm. Now let's talk about reactivity of enteroviruses to chemical and physical agents. This virus can resistant to low pH value, indicating this virus can replicate in our guts. And also, the virus can resistant to our digesting enzyme, so this is why this virus can replicate in our enteric system. And this virus also resistant to alcohol and ether. But this virus can be inactivated by chloride and also by heating or drying. The genome structure of these enteroviruses contain a very long RNA, contains 5' untranslated region and also 3' untranslated region. In the middle are the coding region, which will be translated into a long polypeptide and later on will be caught by viral protease or cellular protease and into 11 mature viral proteins. For example, 2A and 3C are viral proteases, and 3D is the viral RNA polymerase, which is responsible for viral RNA replication. The 5' untranslated region, also called 5' non-coding region, 5' NCR, in the virus genome, is very unique because it is highly structured. As you can see in the slide, the 5' end is the clover leaf structure, which is responsible for viral RNA synthesis. The rest part are stem loop 2 to 6, forms so-called internal ribosomal entry site, IRAS. So unlike cellular translation machinery, it's cap-dependent translation. And here, virus use this IRAS to do the IRAS-dependent translation. Now let's move to the second part about how viruses replicate in the cells. So it's the life cycle of enteroviruses. The first, the virus will attach to its receptors. Different enterovirus got different receptors. After entering to the cells, the virus will uncoat its capsid proteins and let its genomic RNA to be released. And then the RNA can serve as messenger RNA to be translated into 11 viral proteins. After that, the virus will do assembling and to include the capsid as well as the viral genomic RNA and become the progeny virus. And during the replication, the cell will die because of apoptosis, so the virus got a chance to release. This is the life cycle of enteroviruses. So who is at risk for enterovirus infection? In general, it's children. but sometimes adults also can be affected by enteroviruses. For example, poliovirus can infect children as well as adults. And for neonates or very small children, they are at high risk to be infected by Coxsackie A viruses, Coxsackie B viruses, and numbered enteroviruses such as EB71, which can cause very severe diseases. When enterovirus is more active, it's talking about seasonality of enterovirus infection. In general, it's in summertime, but in Taiwan, from April to October, we always can isolate enteroviruses, indicating enterovirus activity is quite common during spring, summer, and even fall. So it's quite long for the whole year. Regarding the transmission of enteroviruses is fecal to oral route transmission, indicating the contaminated water, food will be the resource for enterovirus infection. Of course, the infected children, they will spread the virus to each other, is the major cause of enterovirus transmission. How does enterovirus cause disease? Talking about pathogenesis of enterovirus infection. This virus enters our body through our gut. And we got gut immunity, so sometimes it's okay without any symptom. But the thing is, when our immune response is not that good, the virus will penetrate the gut and go to other organs. If the virus go to the skin and cause harmful and mild disease, which may be fine, but the thing is, when the virus attacks our heart or our brain, then cause very severe diseases, such as myocarditis, encephalitis, meningitis, paralysis, and even death. So it really depends on the targets of the virus. If go to the very key important organs, then cause our body trouble. This table shows the disease caused by enteroviruses. As you can see here, one symptom may caused by different kinds of enteroviruses. And one specific enteroviruses may cause different symptoms. For example, such as encephalitis or meningitis can cause by polioviruses as well as EB70 or EB71. And which kind of virus can cause harmful and mild disease by Coxsackie A viruses, A6, A9, A16, and EB71. And on the other hand, so we can see EB71 can cause harmful and mild disease, and also can cause encephalitis and even death. So indicating if we do the clinical manifestation from clinical symptoms, it's so hard to know the patient got infected by which kind of enterovirus. So this is why we should do laboratory diagnosis, do some laboratory work to know which kind of virus infect the patient. Okay, now let's move to the sixth part of today's lecture, is laboratory diagnosis of enteroviruses. First, I'd like to emphasize again why laboratory diagnosis is so important. As mentioned, from clinical manifestation, it's so hard to know which kind of enterovirus infect the patient because the symptoms are always so similar. So laboratory diagnosis to do experiment to prove which kind of pathogen caused the disease is very important. And first, I'd like to talk about the specimen collection. The clinical doctors will collect the specimen either from stool or rectal swab or throat swab or CSF because of brain infection. And then the specimen will be delivered to laboratory. Because the virus is live virus, so we had better keep all the specimen on ice during transportation. Otherwise, the virus will die. After the specimen are transported into the clinical laboratories, what should we do in the laboratory? We will do three kinds of tests to to identify which kind of enterovirus infect the patient. The first one is cell culture, and then we use immunofluorescence assay to identify the specific enterovirus. The second one is RT-PCR or real-time RT-PCR, and the third one is plaque assay. I will introduce in detail for these three methods in the following slides, and we will also show you how to do the experiments in detail, every procedure and every steps in the next section.